The Delta variant of COVID-19 has spread to 132 countries, appearing to be more transmissible and severe. According to the World Health Organization, the Delta strain is the cause of most new infection cases in all parts of the world. While current vaccines in use are effective against the virus, there still are questions as to how much protection they will continue to provide as the strain gets fitter and faster. And as the cases continue to surge, it is critical to help those who are infected recover quickly and soundly with effective treatments. So how far have vaccines and therapeutics come, and is there a way to make them effective against future strains? For this, we connect with Arirang, Professor of Virology at the University of Western Ontario, who is a globally renowned first-generation researcher of molecular virology and won the Presidential Award and the prestigious Hua Medical Prize, among many others, for his contribution. We also connect with Kevin Morris, Professor of Pharmacology and Medical Science at the Menzies Health Institute, Queensland, at Griffith University. He is also the Deputy Director of the Centre for Gene and Cell Therapy at the City of Hope Beckman Research Institute. Very warm welcome to you both. Thank you so much for joining us today. And well, let's start with you, Dr. Kang, joining us from Ontario. Well, you founded the company Sumagen, which is a company specializing in the development of new biopharmaceuticals. And the vaccine platform you are using is a livestock virus called the vesicular stomatitis virus. Forgive me if I didn't pronounce that correctly. Could you explain more about how this virus platform works and how it builds immunity effectively?
Certainly. There are about 7 different strategies to make vaccines against the various diseases. And out of those, 2 strategies use so-called vector-based vaccine. And there are 2 different vector-based vaccines. One is replication competent, which is what we use, vesicular stomatitis virus, or VSV in short. And also there's a replication incompetent virus, which is like adenovirus that AstraZeneca uses, the chimpanzee adenovirus, and Johnson & Johnson uses the human adenovirus 26 and so on. So those are the vector-based vaccines. We can now make these viruses as non-disease causing so that we can introduce mutations so that the viruses can carry the genes of interest. In COVID-19 vaccine case, we put the spike protein gene into the vesicular stomatitis virus and then make a vesicular stomatitis virus to replicate inside of the vaccinated people and express those spike proteins and come out it, and it can trigger immune responses to make neutralizing antibodies. That's the strategy. So VSV is a very attractive vector because it has a very wide host range, and meaning that it uses LDL receptor, it's a low-density lipoprotein receptor, so that all mammalian cells do have that receptor. Therefore, VSV can infect all mammalian cells, including humans. So we developed the platform technology using this VSV as a platform to carry the genes of interest to make vaccines. And the VSV platform technology can produce the large quantities of vaccines, and it can trigger immune responses to make neutralizing antibodies much stronger than the many other strategies that people are using. So replication incompetent virus, you have to add as much as 50 billion virus particles. But in the replication competent virus, like a VSV, we can use 100 times less. However, the induction of neutralizing antibody is at least 10 times more than the other strategies. So we have seen from our vector system that the very strong immune responses have been induced, and we have done all the protection experiments showing that is highly capable of preventing the viral infections with this VSV-based vaccines.
Well, that is very encouraging to hear that there is high efficacy rate that you're seeing with the trials that you've had so far. And well, of course, there are efforts to develop treatments for those who have inevitably fallen ill with the coronavirus, of course. And well, Dr. Morris, traditional antivirals, they tend to reduce symptoms and help people recover a lot faster. But your technology uses an RNA technology called siRNA or siRNA, I suppose. Please, can you tell us how this gene silencing technology works?
Right. Yeah. So RNA interference was discovered in the late 1990s in plants, and it's essentially a molecular pathway inside all of our cells, human cells, primate cells, that with a designed siRNA, a double-stranded RNA, it can target an mRNA and degrade it. So what our technology does is we have developed a nanoparticle formulation that contains these small interfering RNAs. They go into the lung cells that are infected with the virus, and they specifically kill the virus. They target it and cut it into pieces. It's the actual only therapeutic that's targeted to the actual virus. And so it uses a pathway that's already endowed or imbued in human cells. So we're just taking advantage of what's already there, and we're priming the cell to kill the internal virus.
So what sounded quite fascinating was that you're directly attacking the virus in the system,
Right.
and that was quite new, I suppose. Although I'm, of course, not a therapeutic specialist, but well, Dr.
Yeah, there's two approaches that target the virus. One is RNA interference, and the other is CRISPR. CRISPR is a lot bulkier of a system, but RNA interference works very well. This is a perfect utility of RNAi.
And well, Dr. Kang, you're now holding clinical trials of COVID-19 vaccines, the ones that you're developing with Sumagen, and your technology has been found to be quite effective against the South African variant. So how have you been responding to the new surge of variants, including Delta?
Certainly, we are working on the Delta viruses now, but we have tried to immunize animals with our vaccine and challenge them with the Alpha variant, which is from UK, Beta variant from South Africa. And we saw that the good protection against those variants of the SARS-CoV-2.
And Dr. Morris, I would like to hear how you've been responding to the surge of new variants like Delta as well in terms of developing your treatments. Do these mutations affect the development of treatments for COVID-19?
No, our approach uses survivorship bias. So we designed our therapeutics to target all the beta coronaviruses. So any coronavirus that pops up, our therapy works against it. So like, for instance, it works against Middle Eastern Respiratory Syncytial Virus Syndrome, MERS, which is really deadly. It works against SARS-CoV-1. It works against SARS-CoV-2, and probably any other SARS virus that pops up. The mutations are irrelevant in the context of what we're using.
So this would effectively help you respond to future variants then?
Yeah, basically our approach is a therapeutic. So if people are sick and the option is take our approach, we haven't proven it works in humans yet, but we've done it in mice and cured them. But our approach, if it ever gets taken into clinical trials and works, would basically be something that you come in the hospital, you'd be given this therapeutic versus intubation or oxygen. And it targets the virus and knocks the virus down. It also could be used with dexamethasone, which is a current therapeutic that's used to knock the immune system down. So it's just a therapeutic. It would work similar to Tamiflu, which is used for flu viruses for the first week, first week of infection, where you're really, really sick and highly infected, where we had a lot of virus going on.
And Dr. Kang, now
9 days.
Both the South Korean and Canadian governments are backing your research and developing your COVID-19 vaccine. And so I'm curious, what are your plans? I mean, what does your timeline look like?
All right, we have received the Canadian government grant. It's from Canadian Institute of Health Research in the last year, actually spring of last year, so that this is for the development of the vaccines. And then this year we received the funding from Korean government for the animal toxicology studies and all the follow-up studies to make this vaccine to go into human clinical trials. So we have received the funding, research funding from both governments. And we are working diligently to make our vaccine to go into clinical trials.
And the question is, Dr. Kang, how available and then how scalable the production of your COVID-19 vaccines will be?
Bacillus pneumonitis virus is RNA virus. It actually replicates very, very efficiently. And you can get 1 billion virus particles within about 10 hours of incubation. So it's highly scalable, and you can make hundreds of millions of doses of the vaccines relatively easily. And so we are looking for the CMO, contracted manufacturing organization, who can produce these vaccines for us so that we can do the human clinical trials, as well as we can go on and then have this vaccine available to prevent infections of not only the Wuhan strain, the original SARS-CoV-2, but also many variants coming out from so many different countries. And we are currently testing our vaccine against all the alpha, beta, delta, and lambda variants from Peru. So we will have these data soon. But looking at the neutralizing antibody titers, we are getting, as I said before, 10 times more than what's been reported. So that the level of neutralizing antibody is highly significant for the protection of the people from infection of these variants.
Well, it's incredibly encouraging to hear that it is very scalable and very available
Yes.
for mass production. And
Right.
Dr. Morris, much of the focus until now of national COVID response plans, they've tended to be on inoculating the populations throughout the world. And while it is, of course, very important to roll out vaccinations, it's just as crucial to develop effective treatments that can cure people who have fallen ill with the virus. So what are hospitals mostly using at the moment, and how important is it to broaden treatments from antiviral drugs like remdesivir to biologics based on monoclonal antibodies?
Right. So essentially, we're going to need therapeutics because people aren't all going to take the vaccines. You know, you can have 1 million vaccines and they can be super safe and super effective, but not everyone's going to take them. In fact, if you can get over 50% in some countries, you'll be really excited. My understanding, I'm not a clinician, but my understanding from clinicians I talked to, the current therapy, at least in San Diego, was essentially treat people with dexamethasone to knock down the immune inflammation in the lungs. The problem when you do that is that the virus then pops up, and they had no way to control the virus. So they use remdesivir. Remdesivir is not terribly efficient. It's just barely significant because it's targeting a whole RNA pathway, right? So it's not targeting the virus. And the monoclonal antibodies they do work, early on, but again, you have resistance that's arising to the various monoclonals. So then you have to reformulate those monoclonals. And they're not really targeting the viral genome. They're just targeting the spike and trying to inhibit the virus in the blood from further infection and to neutralize it. So those are the approaches that we're using. But it's really been a push for vaccines because that is, the cheapest, easiest way to try to control with herd immunity the viral infection. But it's not going to end. COVID's not going anywhere. You can vaccinate the entire planet. The virus is going to be here forever. It's now part of our fabric. It's not. I don't think we'll ever get rid of it.
And of course, treatments can't be complete cures, but of course they can definitely save lives. And well, what does your time frame look like, Dr. Morris? When do you think your treatments will become widely available?
Well, of course, like anything, it depends on getting funding to do a phase 1, phase 2, phase 3 trial, which we haven't secured yet. And like everyone, all the money's going into vaccines right now, and there's very little into therapies. This is exactly what happened in 1984 with HIV. It was this big push on vaccines. And then later on, they hit AZT and figured out, hey, we can use a therapy to control this. And I think, it'll start, the Biden administration is starting to put some resources into this and realize that the vaccination is not the panacea that they think it is, because people won't comply. So there's nothing you can do about people's rights to not take a vaccine. So yeah, if we ever get funding for it and get some industry input, let's get some people to put some money in. It's very cheap to do this. But I'm on to working on other things too. This is just one part of our group's efforts. and we don't have any funding for it, so we can only do so much.
Well, it looks like a lot of countries where vaccines haven't become so readily available definitely need to look into therapeutics, and hopefully we will see more developments there in terms of funding, which is of course crucial. And well, Dr. Kang, now how can we ensure that the next time a new type of infectious disease occurs, we can increase our ability to quickly respond to it?
Right. If you have a platform technology which can be applied right away, if there is a new emergence of the new disease, we can move fast. And now there are some unsolved problems, and then all the emerging viral diseases in the last half century, we do not have vaccines against all of those viruses. So we did make vaccines against many of those viruses. And if it really recur, then we can use our vaccines to prevent that. In addition, if there is a new virus comes out, and we have to be prepared to make a vaccine as quickly as possible. That mRNA vaccine strategy was remarkably fast. And in 10 months, we had a vaccine ready to be used to prevent COVID-19 infection. And our strategy, our platform, can also be applied to make vaccines against newly emerging viral diseases. So we are prepared to attack those viruses in the future. And of course, we do not know what other viruses will emerge in the future, but certainly this COVID-19 is not the last, and we will have more. So for that, we are trying to isolate many different coronaviruses from bats, because all MERS, SARS, and the SARS-CoV-2 believed to be originated from the bats. And so we have a collection of bats and isolating the viruses from these bats. If any of these viruses emerge as emerging viral disease and creates another pandemic, then we can attack it as quickly as possible. So that's how we try to control this. And our grant, part of our grant was to cover and isolate those new possible emerging viral diseases or viruses from bats.
Well, I'm afraid this is all we have time for today, so this is where we'll have to wrap up the interview. That was Dr. Kang Chirong in Ontario and Dr. Kevin Morris in Queensland. Thank you both so much for your insights today.
Well, thank you for having us.
Thank you.
And to our viewers, as always, thank you very much for watching.